Hi, everyone. Welcome to the Lightning Talks session C. We will be having a panel of seven speakers, and we will have as co-moderator Manuela Runge from the Northwestern University. So if we can start, let's see, we have Abdou. If you want to start sharing your presentations, we will be happy to see you guys. So if you want to open your cameras, and this is a very interactive session. Even though these presentations are just 3 minutes, and we'll try to keep the time very specific, we will have question and answers at the end. So any questions that you may have, please post it on the chat, and Manuela and I will try to do our best to follow every single question, so we can make it at the end. So if you Abdou, are you here?
Yes, I'm here.
Okay, if you want to start sharing, oh no, you're not gonna start sharing. So I'm gonna just before. Okay, so I'm gonna give you a little presentation of you. So Abdou holds, I think he's a very prolific student. He holds 2 master's degrees, one in economics, one in biochemistry, and one in biochemistry by molecular biology. He also holds a PhD in health economics, and currently he is preparing a PhD in immunology and parasitology. The topics that are going to be covered in this doctorate include, but are not limited to, immunoglobulin G and opsonic phagocytic test. So take it away, Abdou. It's all yours.
Thanks. Thanks for being here today. Hi, I am Abdou Fall, and I am a PhD student at the University of Paris. Today, I will present the work entitled Opsonizing Antibodies and the Control of Asymptomatic Malaria Infection in Beninese Children. Why this work? Previous results showed a group of infants in Benin able to control malaria asymptomatic infection called CAMI, and also characterized by a high level of IgG against antigens such as MA1, MSP, and GLOB. So we aim to investigate the ability of antibodies of children from this group to stimulate phagocytosis, compare it to other children by using opsonizing phagocytosis. And also we study other parameters that could influence OP levels such as IgG levels and the combined polymorphism Fc gamma receptor and IgG. An active parasitical and clinical follow-up was realized on 1,500 infants from birth to 18 months. IgG was purified from plasma at 18 months, and we used THP1 and primary neutrophil cells to do the assay, and also these cells we genotype these cells for Fc gamma receptor, and we use inhibition of hemagglutination to determine the GM allotype. As regards the result, first of all, a high level of OP value was found in children able to control malaria asymptomatic infection when using both THP1 cell and primary neutrophil, with a high significant level when using primary neutrophils. And secondly, we found that a high level of IgG against MA1, MSP1, and GLURP-R2 was associated to a high level of OP, And finally, we found 3 combined polymorphism Fc gamma receptor G3M allotype associated to a high OP. The first G3M allotype 5,6,14,24 when using both THP1 and primary neutrophil, and the second G3M 5,6,14,15 when using only primary neutrophil. In conclusion, this study showed that opsonizing antibodies is involved in the control of malaria asymptomatic infection in Benin. And secondly, among all IgG against antigen, only IgG against MA1, MSP1, and GLURP-F2 appears the most implicated in the control of malaria asymptomatic infection. And also combined polymorphism Fc gamma receptor and G3M allotype polymorphism are involved that could involve on malaria vaccine development. Thanks.
Okay. Well, thank you very much, Abdou. We'll have our next presentation now from Victoria. And please remember, if there are any questions, posted on the chat, and this will be followed at the end in a question and answers session. So, Victoria. Manuela, do you want to?
Yeah, so we welcome now Victoria Shelus. She's a PhD candidate in health behavior at the University of North Carolina at Chapel Hill. She's a Fulbright Fogarty Fellow in Public Health and currently conducting her dissertation research in Western Uganda. Victoria's research focuses on understanding behaviors related to infectious disease risk to inform the development of community-level public health interventions. So welcome, Victoria. The floor is yours.
Okay, thank you. So my presentation today is on the inappropriate sale of antimalarials from private sector drug shops in rural Uganda. So in rural Uganda, as well as many other low and middle income settings, drug shops are often the initial point of care for fever. These privately owned small businesses are often unlicensed or run by unqualified vendors, and clients can purchase antimalarials without a confirmed malaria diagnosis. So the purpose of my study was to understand malaria diagnostic and treatment practices at drug shops in Bugoye, which is a rural mountainous sub-county in Western Uganda. In particular, I wanted to know how many clients were tested for malaria before they purchased anti-malaria at the drug shop, and then how many clients who purchased antimalarials without a test actually had malaria. In total, 46 drug shops participated in the study by collecting data from 934 clients who came to their shops requesting antimalarials during a 2-week period. They also collected blood samples that were stored on ice and then transported to a lab for malaria testing using rapid diagnostic tests or RDTs. Among our key findings, we found that it was common practice for family members or friends to come to the drug shop to purchase medications for someone who was sick at home. So 30% of patients were not present at the drug shop. So in those cases, the vendor could not observe the patient or conduct any tests. Secondly, we found that most clients, 60%, were not aware of their malaria status. The 40% who were aware had RDTs that were conducted at the drug shop or at public health centers that had medication stockouts. In addition to many clients purchasing antimalarials without a test, over a third of clients who tested negative for malaria still purchased antimalarials. And finally, of blood samples collected from clients who purchased antimalarials without a test, approximately 2/3, or 65%, tested negative for malaria. These results indicate there's a need for interventions to improve malaria case management practices at drug shops. Earlier interventions focused more on the introduction of RDTs, but now that they're widely available but infrequently used, new programs are needed. Since drug shops are providing a lot of antimalarials, successful interventions to increase the use of RDTs or adherence to the results could improve rational drug use, reduce unnecessary spending, and also delay the development of parasite resistance. Thank you.
Thank you very much, Victoria. So now we're going to move to our next presenter. Is Hongquan Li. He's a graduate student from Stanford University. He has been working on developing scalable high-throughput microscopy solutions and applying them to diagnostics and research in infectious disease, including malaria.
Hi, everyone. Today, I'd like to introduce a high-throughput microscopy solution for malaria. So with the rise of RDT evading and drug-resistant mutations, we think microscopy is still highly relevant for the management and research of malaria. Back in 2019, we have posted about our first imaging system, and since then we have improved the system to make it more robust, more scalable, and better performing. So the imaging system is a fully motorized microscope that supports wide-field differential phase contrast and fluorescence. We use DAPI staining and found that DAPI has a spectral shift for RNA molecules, which results in improved contrast for the parasites. Fluorescence staining also allows us to use lower magnification for improved throughput. With the 10x objective, the system can screen more than 1 million red blood cells per minute and 250,000 at 20x magnification. So you can see some examples, images of parasites and platelets we obtained with the latest Octopi 20x magnification, which is in the middle of the slide. And then on the top right corner, we also showed some results from the early version. So we're still working on improving the classification and quantifying performance. But from the results, you can see that, so those are scanned of 2 high parasitemia samples, and you can see 2 clusters. You can see the clusters of parasites emerge in the scatter plot. And in the video below, we show low and high magnification image pairs, which highlight the reliability and utility of spectral shift for detection of parasites. At 20x magnification in the latest version, and with both spectral feature and morphological features, we expect to reach detection limit below 10 parasites per microliter of blood with 100% specificity. We think the system can be of immediate relevance to malaria research programs and eventually to the clinical diagnosis and management of malaria. Thank you.
Right. Thank you very much, Runge. So for our next presenter, we'll have Lucie.
Hi, everyone.
Lucie, so let me introduce you then. So Lucie is currently a first-year medical student at the University of New Mexico. Prior to medical school, she completed her PhD in Dr. Bryce Chakarikan lab at the University of New Mexico. Her work focuses on the development of novel vaccines on a virus-like particle platform. So take it away, Lucie.
Thank you. Hi, everyone. So my talk today will focus on utilizing the monoclonal antibody L9 circumsporozoite protein epitope for the development of a virus-like particle vaccine for Plasmodium falciparum pre-erythrocytic malaria. So the L9 antibody is a human anti-CSP antibody that was identified by Lawrence Wang and Robert Cedars' group at the NIH. And it's been shown to be potently protective against infection in animal studies. And its epitope is shown here sort of below the diagram of the circumsporozoite protein, and it contains a unique sequence of alternating minor and major CSP repeat motifs that are shown here in purple and red. And so to generate a vaccine using this particular epitope to elicit an L9-like antibody response, our lab utilizes Q-beta bacteriophage virus-like particles, or VLPs, which are these highly immunogenic self-assembling particles that are made of viral structural proteins And they can be used as a vaccine platform to generate an immune response against a very specific and vulnerable epitope. So what we can do is decorate the surface of these particles with the synthetic L9 epitope peptide in a multivalent manner using a chemical crosslinker, and thus generate particles that, when injected in mice, they elicit a specific and robust antibody response targeting the L9 epitope of the circumsporozoite protein. So on the bottom left here, we show the immunogenicity and longevity of the antibody response following 3 immunizations with the L9 VLP vaccine in mice. So the y-axis here is the antibody titer, and the x-axis is showing the number of weeks following immunization. And the response remains high and shows very little decline after over 30 weeks after immunization. On the bottom right, we show that following immunization and subsequent in vivo challenge using mosquitoes that are infected with a transgenic parasite, here we're looking for the development of blood parasitemia several days post-challenge, and this is seen on the survival plot. So in green, the L9 VLP vaccine is capable of eliciting sterilizing protection in 60% of challenged animals compared to control animals. And so we believe this shows that the L9 VLP vaccine is a promising candidate for a pre-erythrocytic Plasmodium falciparum vaccine. And we're currently working on optimizing this vaccine to increase both of its immunogenicity and specificity. Thank you.
Okay. Thank you very much, Lucie. So, okay, I see we are getting some questions over there. So you presenters can start thinking about them, and we'll happy to answer them at the end in our question and answers session. So our next presenter is Caroline Jasmine Abanto Alvarez. She's Peruvian and holds a Bachelor of Science in Biology and graduate student, and she's a graduate student and research assistant from the University Peruana Cayetano, specifically in the malaria laboratory, parasites and vector. I hand it to you, Caroline.
Hello, everyone. I'm Caroline Abanto, Peruvian Bachelor in Biology. I am pleased to present you our study. In front of a Plasmodium vivax infection exists 2 possible outcomes, which become symptomatic or asymptomatic infection. This last mentioned produce silent reservoirs, continue gametocyte production, and promote the continuity of parasite transmission. In Peru, of the new cases detected in the last years, more than 80% have been asymptomatic. In consequence, these infections were considered within elimination strategies of the Plan Malaria Zero here in Peru. Our objective is to determine the factors associated with recurrent asymptomatic Plasmodium vivax infection in a cohort of people who received the radical treatment in the Peruvian Amazon. Furthermore, about the methods, we evaluated 29 communities grouped into 4 areas. The study had a longitudinal cohort from the year 2008 until 2010, from which 302 individuals were selected who had a follow-up for 24 months. The diagnosis of infection was made by microscopy and PCR techniques. Continue with the results. First, 66% of the total of patients suffered at least 1 recurrence. A total of 609 episodes were detected, of which 65% were asymptomatic. On the other hand, the change from symptomatic to asymptomatic recurrences had the higher incidence density among individuals. In the first scenario, with 69 cases detected, whose average change time was 375 days. In addition, early in time, there is an increased risk that the recurrence will be symptomatic. Third, about factors associated, it was then determined that living in Area A2 in urban communities and begin an homologous recurrence were risk factors for all and the last recurrences respectively. In comparison with appearing in 2009, and 2010, and living in Area 4 areas with more isolated communities were protective factors for all underlying recurrences too. Moreover, these curves show that in time, there is a greater risk that asymptomatic recurrences are monoclonal and homologous to the first infection. However, there is a tendency for recurrences to change or remain heterologous with initial infection, and it was possible to check when measuring the incidence density of this condition. You can see this into the second and third scenario with the red heads. In conclusion, factors such as geography, time, and genetic variability are associated with asymptomatic recurrences. The factors associated present an active dynamic of changes throughout the year of evaluation. These changes are reflected in the complexity of asymptomatic infection and their elimination. Thank you very much.
Thank you very much, Carla. An excellent talk. Okay, and now we are moving into our next presenter is Sophie. Manuela, do you want to introduce Sophie?
Yes, our next speaker is Sophia Berube. She's a postdoctoral fellow in the epidemiology department at Johns Hopkins School of Public Health under the supervision of Professor Amy Wesolowski. She works on leveraging malaria serology data as well as parasite genetic data to understand malaria transmission. She holds a bachelor in mathematics from Browder College and a PhD from the Department of Biostatistics at Johns Hopkins. Sophie, take it away.
Hi, so today I'm going to be talking about distinct antibody responses to Plasmodium antigens that reveal candidate biomarkers of the intensity and timing of past exposure to malaria. So currently there are a lot of challenges to malaria surveillance. But most of them are from the fact that that surveillance relies on diagnostic methods that require us to capture active or recent infections. But antibodies on their own could provide information about an individual or a population's infection history without needing to capture current or recent infections, but we don't fully understand which antibodies are associated with different timings and intensities of past exposure. So there have been a number of studies that have attempted to answer this question, and I'm going to focus on data from one of them. This was a study of 479 individuals from three malaria endemic regions in Zambia and Zimbabwe. And as you can see from the plots on the left, the three regions have very different levels of historic and current malaria transmission. So this study characterized these individuals' responses to 500 Plasmodium falciparum and Vivax antigens via a protein microarray. And using information from the 30 most reactive antigens, this study found regional differences in antibody responses, but only among children. So our goal was to expand this analysis beyond the 30 most reactive antigens and to see if we could identify differences among adults as well, given that adults have had just a longer exposure time than children. So we developed a novel bioinformatic pipeline that allows us to rank antigens within each sample by fluorescent intensity from these microarray data. And these sample-specific antigen ranks are used as inputs to a random forest model that classifies samples into their region of origin. And using classification metrics from this random forest model, we can see which antigens are most informative for classification. So we carried out this random forest model, and we actually were able to classify children and adults into their region of origin with high accuracy. And again, using these classification metrics from the random forest model, we focused in on 64 antigens that were very important for this classification process. And it turns out that only 5 of them overlap with the 30 most reactive antigens. So regional differences in antibody response among adults suggest that there could be biomarkers of the intensity and timing of past infections among these 64 antigens that are important for classification.
Great. Thank you very much, Sophie. And now we have our last presenter. Our last presenter is Hannah Markle, and she's from the house. She's from Hopkins. Hannah is currently a master, an MS candidate in molecular microbiology and immunology program at the Johns Hopkins Bloomberg School of Public Health. She works in the Norris lab and focuses on the characterization of overnight foraging Anopheles mosquitoes to understand species-specific patterns and behaviors along with malaria parasite prevalence within mosquitoes. Her next steps include pursuing a PhD degree to prepare for a future career in infectious disease research, where she wishes to continue studying vector-borne diseases to improve public health as a whole. Take it away, Hannah.
Hi, thank you. So I'll be presenting my thesis research in the Norris lab surrounding the comparison of overnight to early evening malaria vector collections in Chilenge district, Zambia. So as you can see, Chilenge is in this blue shaded region in the map here. It's in northern Zambia, and it experiences holoendemic transmission despite vector control interventions. So this warrants the further study of vectors in the area. So to do this, CDC light traps were placed in 24 households overnight over a period of 2 weeks in August of 2019. And they were placed in households lakeside and inland, as you can see in the map on the bottom left. And the results, if you move to the center of the screen, you can see that a majority of the mosquitoes captured were Anopheles funestis, which is expected because this is the major malaria vector in the region. And then we also captured some Anopheles gambiae, which is another very efficient vector, and then some other more understudied vectors that could be contributing to malaria transmission. And then if you move to the top right of the slide, you can see that we'll also be looking into household structures and how these structural components may contribute to foraging, the number of foraging Anopheles indoors. And so based on preliminary data, you can see that a majority of the Anopheles captured were in households with grass roofs. So you can maybe use that to potentially shape interventions related to household improvement. And then if you go down to the bottom of the slide in the middle, we'll also be looking at a blood meal source within mosquitoes. And so out of all of the mosquitoes, 20% of them were visually blooded. And from those, we found that most of the blood meal source was from humans. Some of it was undetectable, and that's likely just due to the host DNA being degraded. And then we also found some unknown blood sources that we'll be looking further into, and then also pig and dog blood. And so this is going to be important to later compare to early evening outdoor collections to determine whether human proximity to animals has any relationship to mosquito foraging patterns and therefore malaria transmission. And then last, I looked at parasite prevalence, and I did this through ELISA assays detecting the circumsporozoite protein of infectious parasites. And out of all of the mosquitoes that were collected, I found that 1.5% of them were positive for parasites. And then all of the mosquitoes that were positive, they were morphologically identified as Anopheles funestis. And I'm currently working on confirming this parasite prevalence by qPCR assays. So just to summarize, Anopheles funestis remains the most abundant vector foraging indoors in Chilenge. Of all species that we collected, Anopheles funestis harbored parasites, and then parasite positivity followed the expected rates for the region. And so I would just like to thank the Norris Lab, especially Mary Gebhardt, for allowing me to use her collections, JHMRI, the Isomer team, and then also the communities of Chilenge.
Great, thank you very much, everyone. So we're now open for questions. So I think, Manuela, if you want to read, what do we have?
Yeah, there are a couple of questions in the chat, and thank you to the speakers who already answered some questions addressed to them. So there was one question, just picking one, but I noticed them, to Caroline from Rahul. Caroline, how do you think your findings should guide elimination efforts?
Okay, well, I think, yes, these factors show us that heterodiversity geography in the Peruvian Amazon demands different elimination plans. For example, here, this area have different socio-demographic characteristics and demands that the persons do need for into the daily days. And this influence the persistence of this asymptomatic infection, especially with Plasmodium vivax. In addition, there is an active dynamics of parasites in a genetic level that you could see how in only 3 years of the follow-up, did that changes into the persons in follow-up the recurrences. And this is generating not only this, the influence in surgeons of resistance to drugs, etc. No? And in relation, it is very complex scenario that goes beyond presenting or not presenting symptoms here into the Amazon. All the areas stay into to the same department, the Iquitos, Loreto, but different types of communities present different characteristics that is necessary to evaluate it for more focusized. I think that is important to do the more these types of research to study, especially into a recurrence, asymptomatic recurrence for Plasmodium vivax.
Great.
Thank you.
Thank you. Okay.
So I have a question for Victoria, if I may just ask it.
Yes, please open your microphone, people that wants to make questions to presenters. Raul, please.
Yes, so Victoria, some parts of the world have in addition to inappropriate prescribing, some parts of the world have a problem with spurious drugs, that is allegedly antimalarial drugs that contain no drug in it at all. Do you have any idea if this is a problem in Uganda and how much?
Yeah, so it is a problem in Uganda, both fake drugs and substandard drugs. So I think the most recent estimate was something around 10% of drugs fell into this category, and it is primarily antimalarials. One of the good things is I'd say this is the primary focus of the National Drug Authority. They're really looking into this issue of the quality of the drugs and not so much of the issue that I was looking into about the case management practices.
And there's a question for you from Lucie on this case management stuff, asking what degree would you predict that these inappropriate sales are contributing to resistance?
Yeah, so I'm not sure on how much these inappropriate sales are contributing as opposed to some of the other factors, but I would think that it is substantial based on just how many people were buying antimalarials that didn't have malaria or had tested negative. And I also think that of some of the factors that can contribute to resistance, this is one of the things that we could potentially modify easier than some of the other factors that may be more related to the parasite itself.
Great. Thank you very much, Victoria. I think we don't have any other questions, or like the other questions that have been asked in the chat, presenters have replied to them. I have a questions for Hongquan, like a very, very, very cool instrument. So I was thinking for the microscope, is what about the power source? Is it something that how is it? How does it work with?
Yeah, so because we use the Jetson, Jetson embedded computer, which consume only 10 watts, we can use the battery for powering the device.
So is this something that can be used in the field?
Yes, yeah. It's actually we have brought this out to the field a few times, and we use a battery brick that's this large, then can power the device for a few hours.
Awesome. Great. Great. Well, actually, is there any other questions? I wanted also to ask Caroline, if you have seen any of these frequencies in your studies related to forest implement, like deforestation or things that are really with the Peruvian Amazon with it.
The study produced during a 2 years follow-up for 2 years. In this period of time, the persons evaluated monthly, and the persons every month to submit with diagnosis with our PCR and microscopy, and for these reasons we could see evaluated the asymptomatic presence of recurrence. In the majority of cases, were the first infections that the person is presented symptomatic with initial infection, but when they evaluated to the rest of the time, the persons for the last part of the years, especially during the 2009 and 2010, the majority of the recurrence are symptomatic. You could see that especially when you this infection are monoclonal, and this type of the parasite, but when evaluated during the years, the variability genetic are changed into the haplotypes, and the variability for the last recurrence are heterologous. This is really interesting how in this period of time that the plants like Pamafro, the affected allow cases into these years, the effect monoclonal that is not suffered for the parasite continue to mutate and generate new types of haplotypes, and this is really interesting.
Great.
Yes.
Yeah, I'm just reading in the chat that these communities are located around only a road in this area, but also yes, this deforestation could be also a factor here. So this is a very interesting field of work and with a highly impacting and relevance medical.
Yes, during these times, these factors like entomology and deforestation that is no more focused in these times, but yes, this is a factor that is influenced now in more recently studies.
Right.
Here in Peru, they work a lot in this.
Well,
yes.
Thank you very much, everyone. Before everyone leaves, we want to share some advertisements for keynotes of Malaria Keynote Symposium in April 10 to 13 in Malaria Confronting Challenges from Drug Discovery to Treatment. And last but not least, we also want you to save the date. April 25th, 2022 will be the World Malaria Day. We'll be looking forward to see you more in future meetings. And thank you very much. Thank you everyone for staying, and thank you very much. It's been a wonderful meeting.